Thanks, Bill. Thanks for the introduction and also for inviting me to this and organizing this very nice meeting. So cells, living cells, right? So they live in a very dynamic environment, right? And they have to continually respond and adapt to this environment. And as you probably know, they have developed several strategies for doing this. One is the bed hedging strategy, which relies on maintaining phenotypic diversity at the population level. The other strategy is to detect the change in the environment and respond accordingly. But perhaps the most interesting strategy is that of prediction and anticipation. Mounting a response takes time, right? And it can therefore be essential or beneficial, if not essential, to mount the response ahead of time and to mount it in time, right? As this Dutch example perhaps illustrates rather dramatically. Now, this idea that organisms can predict and anticipate, right? People often associate that with higher organisms, but it's now clear that even bacteria can do this, right? And so it has been argued that E. coli, the bacterium E. coli, in our gut, in the gut of mammals, sees sugars in a particular temporal order. And in particular, it has been argued that the arrival of maltose is preceded by the arrival of lactose. This is just a result of the metabolism in our body, right? And this means that E. coli can use the arrival of lactose to predict that maltose will arrive. Right? Jesus. Time is up, sorry, talk is over. Okay, I'll just switch it off. I'll keep an eye on that clock.
Yeah, that's actually not such a good idea.
Oh, that clock, no, it's also not, yeah, okay, and then we also have to stop already. Okay, well then you have to warn me at some point. Okay, good. Right, okay, so, so it has been argued that because of metabolism in the host, E. coli sees these sugars in a particular temporal order, right? In particular, it has been predicted that E. coli experiences lactose before maltose, and then can use the arrival of lactose to predict that maltose will come, right? And if this is true, then E. coli, the prediction would be that the expression of maltose is influenced by lactose, right? And this indeed turns out to be the case, right, as was demonstrated in this paper by Mitchell and co-workers, right? So here, right, you can see that when you give the bacterium the sugar lactose, it does not only express the lac operon, but it also expresses the maltose operons, which are necessary to consume the sugar maltose. Right, so clearly, upon the detection of one sugar, E. coli can predict that the next one will arrive. Yes, right. I'll come to that. Right, and and this also can provide a fitness benefit, which is shown here on the right. Right, so the black symbol on the right indicates the growth rate advantage of cells that have been pre-exposed to lactose and therefore can grow faster on maltose. And the control experiment, right, so one is that, right, if you let E. coli now evolve in an environment in which this particular temporal order is lost, right, so if you evolve E. coli now in an environment in which there's no maltose after lactose, then the prediction would be, right, that it can no longer, that it then no longer responds to lactose when it comes to the expression of maltose, right? And so that's what you can indeed see, right? So now the bacterium no longer expresses maltose in response to lactose, at least not as strongly as it did before. And then also this fitness benefit goes away, as you see with this blue dot over there, right? And so this fitness benefit is then lost, right? So this example goes to show that even a simple organism like a bacterium can make predictions. Now, of course, a prediction is only useful if you can make it reliable, right? And so this is then immediately the overarching question of this talk, which is how reliably can living cells, single-celled organisms predict the future? Now, before I continue, I'd like to thank the people who did the work. First of all, my PhD student Aage Tjalma, my postdoc Vaha Galstian, former students Jeroen Goedhart and Lotte Slim, and my long-term collaborator Niels Becker. Now, how should we think about this? Right, so here I have a schematic of an input signal in red as a function of time, which via some sensing system generates an output, here shown in blue, right? And the cell needs to predict from this output what the future input at some later time in the future will be, right? So while the cell needs to predict the future input, it can only sense and remember the past input, right? It can only sense the current and remember the past input, right? And as Bill and others have shown, right, this immediately means that the amount of information that the organism can have about the future, the predictive information, is fundamentally bounded by how much information it has extracted from the past signal. And how you can do this within this information bottleneck method I will discuss shortly. Here is just the basic idea, right? And so, and what's important to realize is that this information bound only depends on the temporal statistics of the signal. It depends on how much temporal structure there is in the signal, right? So today we will also be considering different types of signals, and they will give rise to different information bounds, right? Right, so this bound is really a property of the temporal statistics of the input, not of the system. But how close the system can come to this bound and how much information it extracts from the past signal, that does depend on the design of the system, right? And so today we will be considering biochemical networks, right? So we imagine a cell with receptors on the surface that detect the ligands, which is the input signal here in red, and this receptor drives then some downstream network with some output X, right? And then from the output X, it then needs to predict what the future input is. So this is sort of the general setup, right? And what's important is that how accurately the cell can come to this bound, right? That depends on the design of the network, right? So in order to reach that bound, the cell should extract those characteristics that are most informative about the future input. What the most informative characteristics are, that depends on the temporal structure, that depends on the nature of the signal, right? Whether the cells can then extract these characteristics, that depends primarily on the topology of the network, right? I will make all of these ideas much more concrete as I go along, right? So the general idea is you have a signal with a particular temporal characteristics. Those have features that are most informative. The cell should extract those features, and whether it can do so primarily depends on the topology of the network. So that will determine how close the cell can then actually come to the bound. Now, how much information it actually extracts from the past signal, that also depends upon the topology, but it also depends on the resources that are devoted to making and running this network, right? So, so this network needs receptors, it needs readout molecules, right? So you need to make these building blocks, right? So there's a maintenance cost associated with making these proteins. And then in addition, there's also a cost with running this network, right? These networks are typically driven out of equilibrium, right? And this means that there's just a chemical power associated with just driving this network, right? And these resource costs, they determine how much information the system can extract from the passed signal, right? And as Bill pointed out so beautifully in his book and also in papers, right? This then determines, these resources determine how much information the cell can extract from the passed signal, the passed information, and this in turn will put an absolute upper bound on the predictive information, right? And so this is the general idea. Now, in the context of neuronal signaling, a lot of progress has been made in the code, in understanding the coding of information, in characterizing the relation between predictive information and past information. And Laughlin also has a nice paper on estimating the ATP cost of running this network, right? But quantifying these resource costs for neuronal systems is arguably much harder than here for the cellular systems, right? So we know, for example, for E. coli, how many receptors it has, how many readout molecules it has. We know the growth rate, so we know on what time scale these proteins have to be synthesized. We know what this relaxation time scale is. We know the free energy of ATP hydrolysis, so we can really quantify these resource costs, right? And this means that these cellular systems are ideally suited for studying the resource requirements for cellular prediction, right? So what are the questions then that we will address today more specifically? So first of all, what is the information bound? How can we compute this? What is the design of the optimal system that can actually reach this information bound? How does that depend on the statistics of the input signal? How close can biochemical networks then come to this information bound, right? And more generically, what is the optimal strategy for extracting past information to maximize predictive power, right? So we're going to address these questions for two classes of signals, a Markovian class of signals and a non-Markovian one. So the signals that we will consider today will obey Gaussian statistics, right? And it has been shown by Čechić and others, right, that then the optimal mapping is actually a linear one, right? And we also know that many biochemical networks, and certainly this network, Katja also already discussed that, can be described to a very good approximation via a linear model, right? And that's illustrated here, right? So the output X in the context of E. coli, that would be, for example, Ky, right, is given by this integral of the response function chi and the input signal S plus some noise term, right? And what is important is that this response kernel chi, that determines which signal characteristics are extracted from the signal and are mapped onto the output X, right? And so when we talk about optimizing the network and optimizing the topology, we start by thinking about optimizing this response function. Then we will start with the simplest Gaussian signal that we can come up with. That's just an Ornstein-Uhlenbeck process characterized by this correlation function, right? So a correlation function that has a variance sigma squared s, and that decays exponentially on some timescale tau l, which is the input timescale. So this is a Markovian signal, right? And then we compute this information bound using this information bottleneck method that Bill and Natalie Tisby and others have developed, right? And so the basic idea is that it's a constraint-based optimization. So you maximize the predictive information, which is the mutual information between the output X in blue and the input, in this case, at some future point, at some future time. So that's the predictive information, and you maximize this subject to a constraint of the past information, which is the mutual information between the output and the past signal trajectory, right? And then using that, you can then map out, trace out this information bound, which gives you the absolute upper bound on the predictive information as a function of the past information. And when you do this maximization procedure, you also get an expression for the optimal response function, chi, right? And so when you do this, right, what you then find is that the optimal response function for such a signal is just a delta function, right? And that's not surprising, right? Because a Markovian signal means that all the information on the future is contained in the current signal. There's no additional information in the past signal that's not already contained in the current signal, right? So the optimal network is a network that makes an instantaneous copy of the input signal. It instantly copies the input S into the output X, right? And from previous work, right, we figured that a push-pull network should be able to do this, right? So push-pull network is this motif. It's the most ubiquitous motif in cell signaling. Essentially, any signaling network has a push-pull network, right? And so here in this case, right, it's this ligand binding to a receptor, and then this ligand binding state of the receptor, bound or not, is copied into the chemical modification state of the readout, phosphorylated or not, right? And so this is a copy process, and if you run it fast enough, it should in principle be able to make an instantaneous copy.
Sorry. You've written this, there's no noise that comes along with signal. All the noise comes at the output.
In the way I write it here, yes, indeed.
And that's important for the instantaneous.
Absolutely, absolutely.
Okay. Yeah.
Great question. I'll get there. So, definitely, this is very important. Okay, so right, so in principle, a push-pull network can make an instant copy. So in principle, it should be able to reach this bound, right? And so we're first going to analyze this by just focusing on the maintenance cost. For now, we're first going to ignore the operating cost. I will come back to this later. Then I will also study separately the effect of the operating cost, right? And so, what we're going to do is we're going to basically map out these two quadrants shown here on the right? So these are two of the four quadrants in this book of Bill, and we refer to this beautiful figure as the windmill figure. I don't know whether you're happy with that, but when people in the group talk about this figure, it's the windmill figure. But because basically you also have a quadrant then of predictive information to the utility of information, and then you can connect this back to resources, and then you can make the connection from resources to past information to predictive information to performance, and then close the loop. And here we're only looking at two of these quadrants. Okay, so what are we now going to do first, right? So what's important, right, is that in order to make a prediction, the cell should extract the information from the past signal, right? It bases its prediction, obviously, on the past signal. Right, so we start rather naively, right? So we start by asking the question, does maximizing predictive power, does that actually imply maximally extracting past information? Right, so what we're going to do first, for a given resource constraint, we're going to optimize the past information, and then we're going to ask, how well does that system when it comes to prediction? Right? And so what's important, right? So this is our constraint, right? This is the growth rate which we just keep fixed. It's 1, right? And so for a given constraint, we're essentially going to optimize the ratio of the number of readout molecules over the number of receptor molecules for a given constraint. And then we're also going to optimize over this integration time, tau r, which is basically how fast this network cycles between the phosphorylated and the unphosphorylated state, right? so this is then what we found, right? So we maximized the past information as a function of these resources C, which is just the sum of the number of receptors and the number of readout molecules. That generates this blue curve. And then we just ask, how well does this system perform in terms of prediction? Right, so we maximize the past information with respect to resources. That's this blue line here at the bottom. And then the blue line here at the top is just how well this system then performs in terms of prediction. And what you can see is that it's below the information bound. Right, and it's quite significantly far below the information bound. Right, and so clearly, optimally predicting the future does not imply maximizing the information extracted from the past signal. And if you think about it, it also means that not all bits of past information are equally informative about the future, right? Because if they would, then these bounds would agree, right? Now, then we decided to do something else, right? So rather than maximizing the past information under a resource constraint, we're directly going to maximize now the predictive information under a resource constraint, right? So the blue lines are the ones that I just showed, so nothing has changed there. Now we're going to add the red curve, which shows the maximum predictive information under this resource constraint, right? And what you can actually see is that this system now is already much closer to this information bound, right? So clearly, for a given amount of resources, this predictive power has now gone up. It has increased. Interestingly, at the same time, for a given resource cost, its amount of past information actually has gone down.
Give us a hint about what's in the model. This is stochastic processes happening at this molecular level?
Yeah, so basically, right, so we are in this Gaussian framework, so we basically use the linear noise approximation, and then we can calculate all the required quantities exactly within that framework. Right? So we find that the optimal system is not at the information bound, right? And if you then think about it, it also means that not all bits of information are equally costly, right? We already saw on the previous slide that not all bits are equally predictive. This shows that not all bits are equally costly. If they would, the cell would just select those bits that are most informative, and then again, the system should be able to reach this information bound. And we know that in principle it's able to do that because it's a push-pull network. I will make this much more concrete on the next slide, right? Because now I've basically just shown two key observations. The question now, of course, is how? How does this arise? Right? So why is the system not at the information bound? Right? So this is again the predictive information, is the past information. Again, there's no new result here, right? So the black is the information bound, is the absolute upper bound of the predictive information as a function of the past information. The red curve, again, is the maximum predictive information obtained under a resource constraint. And then the blue line is the line that we obtained by maximizing the past information under this resource constraint. And what we're now going to do is we're going to look at all possible systems that can be made with a given resource constraint, and then we're going to ask how do they behave in this plane, right? So that's shown over here, right? So let's just focus on this green area, which corresponds to a resource cost of 10 to the 4 or less, right? So again, C is this number of receptors and the number of readout molecules, right? And so here in this area are all possible systems that can be made and that obey this constraint, right? And then the red dots, right? Those corresponds to the system that indeed then maximize this predictive information under this constraint, right? So they make up this red line that I showed before, and the blue dot is the line that maximizes this past information under this resource constraint, and these blue dots together form this blue line, right? And so 2 observations, yes, a lot of questions, good. So let's start there.
I have a quick question. In the red curve, there's a kink around like 2 bits and 2.5 bits. It's like a short transition. Is that a real thing or where did that come from?
The one there with the black dot.
No, like there's like a sharp transition where one's like a corner for the red curve.
There, yes, yes, okay, I can talk about that later. Let me first explain this, and then I can come to that, yeah. No, so here we deliberately take it out. We're going to address that on 2 slides from now. Yeah.
Sorry, could you just remind us what the definition of past information, right?
So that's the mutual information between the output X and the past signal trajectory. And the predictive information is the mutual information between the output and a future point at an input signal at a future time into the future. At a point into the future, yes.
Past information is the same as I-past.
That's I-past. Yeah. Right? So, okay, so the 2 observations that stand out here, right? So one is that indeed a system can be at the information bound or very close to the information bound. Right? That's one important observation, right? Because I argue that push-pull networks should be able to reach the information bound, and this is showing that indeed they can, right? But the second observation, of course, is that the system can increase both the predictive and this past information by moving away from this information bound, right? And so the question, of course, then is, right? So why or how does that come about, right? And to answer that question, we basically look at the design of the system along this iso-cost line, right? So this green line is an iso-cost line where the sum of the receptors, where the sum of the number of receptors and readout molecules is a constant, right? And so we're going to look at the design of this system along this front, right? And then we look at the integration time, tau r, over the correlation time. So the correlation time, that's the correlation time associated with receptor ligand binding, right? There's a time scale for receptor ligand binding and unbinding, that's tau c. And tau r is basically what I said before, right? Is how fast the system cycles between the phosphorylated and the unphosphorylated state, which is the time scale on which the system has memory about its past. So that's really setting this integration time, right? And then, so that's the solid line above, and then the dashed one below is this ratio of the number of readout molecules X over the number of receptor molecules RT, right? And what you see is that along this front, both increase, so clearly there's an optimal integration time and an optimal ratio that maximizes this predictive information. And there's another optimal integration time and an optimal ratio that maximizes this past information. Now, how can we understand this qualitatively, right? So it should be recognized, right, that each readout molecule is either phosphorylated or not. So it provides at most 1 bit of information about the concentration. But that information is relayed via the receptor, right? And more concretely, right, so it's the state of the receptor that is copied into the chemical modification state of the readout, right? And so what this means is that, right, this information from the concentration, the input, to the output is relayed via the receptor. And this is important, right? Because to maximize this predictive information, the cell should make more measurements about its input, right? And so that means that the number of readout molecules, X, must go up, right? I mean, if you want to raise this bound, the number of readout molecules must go up. But if you have a constraint, it means at the same time that the number of receptors must go down, inevitably, right? Otherwise you don't obey the constraint. So the question is, how can you preserve the number of measurements at the level of the receptors when you do this, right? So if you increase xt, you have to decrease RT, right? And you would think that then you decrease the number of measurements at the level of the receptor, right? And in an optimally designed system, the number of measurements at the level of the receptor should equal the number of measurements that you store in the readout. So how can you do that? Well, that's of course the mechanism of time integration, which allows the system to take more measurements per receptor, right? That's really what time integration is, right? And to really drive this point home, right? So here we have, for the sake of illustration, a scenario with 2 readout molecules, 2 receptors, right? And so in this case, there are 2 readout molecules that can store information about the environment. But if you now trade one receptor for a readout molecule, right, then you have one receptor, 3 readout molecules, and now you can store 3 concentration measurements. So you go from 2 to 3. Right? That's really what time integration allows you to do. It allows you to use these receptors more efficiently. Right? And this allows you to increase the total number of measurements. But there's of course a price to pay. Right? And the price to pay has to do with this integration time. Right? So here in this example, I've increased this integration time from 1 times this receptor ligand binding correlation time to 3 times this receptor ligand correlation time. And this is important because if I would not have done that, then the sampling interval of this receptor, so the time in between the readout receptor interaction, would go down. Right? If you keep tau r constant and only increase the number of readout molecules, your receptors will be sampled at a higher frequency. And if you sample them at a frequency that is higher than that set by the correlation time, right, such that the sampling interval becomes shorter than this correlation time, you basically start taking redundant measurements. Right? You take just redundant measurements of your receptor. Right? And so to avoid taking redundant measurements of the receptor, when you increase xt, you must also increase the integration time. All right? And this is basically what we, in this paper, call this optimal resource allocation system. In an optimally designed sensing system, the number of readout molecules, x, should be given by this expression. And it has a very intuitive understanding, I think. Right? So 1 plus tau r over tau c, that's the number of independent concentration measurements at the level of the receptor. If you assume, as we do here, and which is not entirely obvious, that the receptors take these measurements independently, then the total number of measurements stored at the level of the receptor is simply how many measurements you have per receptor, 1 plus tau r over tau c, times how many receptors you have. And in an optimally designed system, that number of measurements you take at the level of the receptor should equal the number of readout molecules that store these measurements. Right? And so, this explains qualitatively that when this integration time, that when this ratio increases, also this integration time must increase. But now you can see that the trade-off arises, right? Because when you increase this integration time, it means that these readout molecules increasingly store states in the past. They store ligand binding states in the past, right? And they are less informative about the concentration that the cell wants to predict, right? And so basically what you get is a trade-off between, on the one hand, the fact that you can increase the number of measurements by increasing this ratio and increasing this integration time, while on the other hand, making these measurements less predictive, right? And that's illustrated here in this panel on the right. So this blue-purple line, that's just the number of readout molecules per resource cost. That just increases. That just shows that as you increase this readout to receptor ratio, you can take more measurements, right? while the dashed line shows that this predictive information per measurement then actually goes down, right? And this interplay gives rise to an optimal ratio indicated in red, corresponding to that red point over there, that maximizes this total predictive information. Right? So what we see is that this optimal system phrases a trade-off between bits that are most predictive and those that are the cheapest. Right? That's really the take-home message here. So now about the power, right? So far I only focused on this. Now we're going to include this power cost, right? And when you do that, then you see that not only this predictive information comes down, but also that it moves slightly away from this information bound. And I think that can be understood very easily, right? Because to be at the bound, this integration time should be 0, because then you make these instantaneous copies that are most informative, right? But that means that you just run this cycle like crazy, and then this operating cost simply diverges, right? And so this operating cost means basically pushes this integration time to larger values, and that moves the system away from this information bound. Right, so the operating cost moves indeed the system away from the information bound. Okay, so this was all about these Markovian signals modeled by an Ornstein-Uhlenbeck process. Now we're going to look at non-Markovian signals, right? And I start here by showing a movie I think that you made, Katja. Right, and so this is about, this illustrates this idea that many organisms, right, they navigate their environment with some directional persistence, right? And that means that the statistics of this signal becomes non-Markovian with memory as generated by their own motion, right? And as Katja already discussed in her presentation, right, is that for this system, this integration kernel has this bilobed structure, right, as was measured first by in the lab of Howard Burke, right? And so one question is, why does this kernel have this particular shape? Well, this has been discussed widely in the literature, right? One is that it has to do with the dynamic range, right? So this ensures that the KYP concentration is always within the motor working range of the motor. Clark and Grant, they pointed out this trade-off between running fast in a gradient and staying near a peak, right? So the basic idea is that if you want to stay near a concentration peak, you actually want to have a negatively lobed kernel, but if you want to move rapidly over a gradient, you want to have a positively lobed kernel. And then sort of the global optimum between the two gives rise to this bilobed kernel. And then Massimo, right, Massimo Vergasola and Solani, they have this idea of a maxi-min strategy, which is basically a twist on the same idea, which is that this kernel is the one that performs best in the worst scenario, maxi-min, right? So again, a negatively lobed kernel does very well on staying near a peak, but a positive one does extremely badly because it actually drives the system away from the peak. So yeah, right, and the positively lobed kernel, that does not allow the system indeed to stay near this peak. So the maxi-min is basically the one that does the best on the worst. But I would say that both still involve some sort of trade-off between staying near a peak versus climbing a gradient, right? And whether that precisely gives rise to a zero integral peak is not so obvious to me in general. Moreover, bacteria, they consume food continuously, so whether this idea of a maximum, a stable maximum, really ever exists, that you can also argue about. Wonder about, right? And so we have sort of a different perspective on this, which is that it has to do with prediction. Right, so how are we going to address this, right? So we're going to modify the dynamics of our signal, right? So it's now basically a particle in an underdamped particle in a harmonic well, right? So L is again the ligand concentration, its change in time is V, so V is the change in the ligand concentration, and that's basically given by the bottom expression shown over there, right? And and then the first question that actually arises, and this is very important, is what does E. coli want to predict? What should it predict? Should it predict the future value? Should it predict the future change? Or a combination, right? And so basically we decided to embrace the dictum of Howard Burke, which Katja also already explained, right? So Howard Burke always liked to say E. coli is an optimist, right? So it expresses the idea that if the concentration is now rising, if life is getting better, then I should just continue going, because then probably life will remain good, right? So that's this idea of E. coli as an optimist, right? And so it sort of expresses this idea that what E. coli cares about is really the change in the concentration, whether that goes up or goes down, and not the value as such. So this is the argument that we actually put in our manuscript. We also, because we believe this argument, I think this is sort of true, and Howard Burke understood this organism better than anyone else probably, so it's also, for that reason, maybe true. But there's also another argument that I discussed that I brought into the group that we didn't put into the manuscript, which is, what does it mean to actually measure a concentration, right? So suppose E. coli would measure a concentration, and it measures that the concentration is 0.8 micromolar. What does that mean? Okay, if E. coli knows that the range of possible concentrations between 0 and 1, then 0.8 is high. But if the range is between 0.5 and 500, then 0.8 is low, right? And then you can say, well, okay, evolution has taught E. coli what the range of possible concentrations is, right? But then, then there's also this fact that E. coli lives in many different environments, our gut, the soil, water, right? And in all of these environments, this range of possible concentrations is probably going to be very different, right? And so knowing then a concentration value is probably not of much use. Okay, this can be debated. Maybe this is a point of discussion. We didn't put this in our manuscript, but I think that these organisms really care about the change in the concentration, right? So that's what we're going to assume, right? So organisms climbing gradients need to know the change in the future concentration rather than their value. And then you can say, well, are you now not tricking us, right? Because we already know that this kernel takes a temporal derivative. So what are you now really saying here? Are you not saying something very obvious? Well, I would like to argue that the converse, namely the fact that it needs to predict a change, does not automatically imply that then the optimal strategy is to only measure the derivative. Because in general, the optimal network to predict, the optimal strategy to predict a future change actually is a system that combines the current value, delta L, and the current V, and the current change, right? And that is because in general, the concentration value can provide information about what the change in the future is going to be. So in general, the optimal system to predict a future change is a system that combines a current value with a current derivative. But now you can ask, okay, what determines then the values of A and B, of these coefficients, right? And what you can show for this model is that the optimal value of the coefficient A, that depends on how much the current signal value is correlated with the future change, while the optimal value of B is determined by how much the current change, delta V0, is correlated with the future change, right? And this makes sense, right? If the current value is strongly correlated with the future change, right, then you should use the current value in your prediction, right? And then A should be non-zero. Now, the non-trivial point, I think, is that what you can show in this model, and I think it holds more generally, right, is that when the range of background concentrations is large, meaning larger than the typical concentration change over this time scale tau v, which is set by the run time, the run duration, right? Then what you can show is that then this cross correlation goes to zero. And then the optimal kernel is a perfectly adaptive kernel, right? And so, so to summarize this, right? So we start from the premise that E. coli wants to predict the future change. That is what we assume from the start. Then we say that in general, the system should then use both the current value and the current change. But when the range of possible concentrations it can experience is much larger than a typical concentration of a run, then this coefficient A, then this cross correlation goes to zero, right? And you can think about this, right? So this is basically a particle in a harmonic well, right? If this harmonic well is very, very large, very, very wide, and if the runs or sort of the changes in direction happens on scales that are much shorter than this width, right, then basically sort of the correlations that you have between the velocity here for x is positive will be the same as here big X being negative, and so then the cross correlations become zero, right? And we know that E. coli can measure concentrations over a very wide range, over orders of magnitudes, basically set by the dissociation constants of the active and inactive state of the receptor, which has been measured. So it can really measure the concentrations of orders of magnitude that are typically larger than the typical concentration change over a run. Right? So basically we argue that this kernel is a perfectly adaptive kernel because E. coli wants to predict the future derivative. That's really what we say here. Yeah.
A dumb question, but when we think about chemotaxis, we think that E. coli is interested in relative changes.
Yes.
So how does that fit into your scheme? Does that explain why the absolute value matters?
No. So it's more important that it's the change rather than the relative change. Yeah. Yeah, no, so I think this relative change probably changes the overall gain, but does not change this conclusion as such.
And I didn't really understand why the value matters, as opposed to just the derivative.
Right, so if you think about a particle in an underdamped harmonic well, if this well is relatively narrow, right, and then it has a velocity going down, right, it's basically going through the bottom of the well, shooting up, and then it will turn. And this creates correlations between its position now and its future velocity. Whereas if this well is very, very large and it changes direction before it reaches the bottom of the well, then this correlation disappears. This is really this idea, right? So if you have this very large well and you start here and you turn before you even reach the bottom, yeah, then this cross correlation between position and future velocity goes to zero. That's really what's going on here. Yeah.
I understand why you chose this model of dynamics, because you can calculate everything. But if I, for example, compute the correlation functions, and I just look at them, and you brought up the example of, well, I'm swimming, and so that induces correlations. If you just showed me the two I mean, if you imagine that there's some landscape of concentration versus position, and you give the usual trajectories, what does the correlation function look anything like this? I mean, it's important here that there are two timescales, right?
Yeah.
What are those two timescales for the swimming bacteria?
Yes, exactly, right? So, so basically you're asking how relevant is this model sort of in a typical, or what do you really assume here when it comes to sort of biology? Is that, may I phrase that question that way?
I would be gentler because I'm sympathetic to using simple models.
Yeah, no, yeah, but we worried about this a lot. So
it seems like the argument you're making is that okay.
Yeah, so we really thought about this, right? So, so as an example, we look at exponential gradients, but this is because exponential gradients are also used experimentally a lot. As such, this is of secondary importance. What is important, right, is this is also what you guys have shown, right, is that when the gradient is very, very shallow, the dynamics, to a very good approximation, is the same as that in the absence of any gradient. Right, so the cells, they swim as if they swim in a constant background, right? And and then the phi is this change in the concentration, right? So that's dL, L is the ligand concentration, dt, right? And then that for this exponential gradient is simply GL by the average concentration times Vx, which is the velocity in the direction of the gradient, right? And what these gentlemen over there, right, measured, right, is that this correlation function of the velocity in the direction of the gradient, that to a very good approximation is given by this exponential form, right? And this is precisely what our model also gives in this limit, which we argued is biologically relevant, namely this limit where the width of this concentration potential is much wider than the typical concentration change over a run, right? And if you then indeed take this tau v in this model to be equal to the tau vx of what you actually measure, right, then our model precisely describes the statistics as measured in that paper, right? So, and we argue later.
The other time scale is the time it takes for the absolute concentration to change by of order its standard deviation.
Yes, but if
the standard deviation is very large,
yeah,
it takes a long time. Okay.
Yeah. So what we argue is indeed what's important is how the concentration changes on the timescale of a run, let's say, right? Because you also want to make predictions on the timescale of the run duration, right? So so really what you would like to know is whether the concentration goes up or goes down over a time tau equal to the run duration into the future, right? And so for us actually, right, because for a long time we really struggled, right? I mean, so it's easy to make sort of a model, right, as you often have, and then you start wondering, okay, you know, how should I map this model onto reality, right? And so for a long time, we were basically sort of stuck with this, right? Because if both coefficients matter, then everything becomes very ugly, because what these optimal coefficients then are depend on your forecast time interval, right? Because this ratio varies with the forecast time interval, and so then everything becomes very ambiguous, right? But if you indeed accept this premise that E. coli needs to make a prediction of the change, and if you then also accept this observation that it can respond over a range of concentrations that is very large, then you find that in the optimal system, A goes to 0, and then that it basically measures the derivative only, because that's optimal for predicting the derivative. So this is basically what we argue, right? E. coli wants to predict the derivative, and the optimal way to do that is to just measure the current derivative. That's what we say. Right, and then what's shown here is that in this regime of shallow gradients, this model, it's simple, but it can describe the statistics as measured experimentally. Okay, so basically what we did then, we looked at the chemotaxis system, right? It has this push-pull network. It also has methylation and demethylation of the receptors. We actually realized that here the receptor ligand binding noise is very, very small. Right, so receptor ligand binding is so much faster than phosphorylation and methylation that here we decided to take tau c to 0, this correlation time to be 0, and then take a constant integration time. And then vary the ratio again of receptors to readouts and the methylation time. So these are now the two key optimization parameters. And then what we then find is this result shown over here, right? So again, black is this information bound, which gives the absolute upper limit on this predictive information as a function of the past information. These green lines are again the ISO cost lines, and then the red line shows the optimal system that maximizes this predictive information subject to a resource constraint C. Right? And what you can see again is that the optimal system is not at the bound, it's markedly away from the bound. Right? And then, of course, again, the question is why? Another interesting thing to note is that before we had these shapes like this, now it actually goes like this. You can also wonder why that is the case. Right? So before we had that the predictive information at some point goes down, but also the past information goes down. Now we have that the predictive information along this ISO cost frontier goes down, but the past information continues to rise. That's maybe another puzzle you may have. Right? And again, to answer that, we looked at the design of the system along this ISO cost line. Right? And the key parameter to look at is then shown here. That's the methylation time. So that's the time scale of receptor methylation and demethylation, right, which is responsible for the adaptation. Right? And what you see is that along this ISO cost line in this direction, this methylation time increases. But what you can also see is that this optimal methylation time that maximizes this predictive information, which corresponds to these red dots, that actually goes down when this resource constraint is relaxed, right? So you can see that, for example, for the dashed line, for C is 10 to the 6, this optimal methylation time is shorter than the optimal methylation time for C is 10 to the 2. Right? So this optimal methylation time is non-zero, but it becomes smaller and smaller as you increase the number of receptors and readout molecules. Right? And well, the way to understand this is that, right, really what this system does, right? So Katja explained that already, right? Is this takes this temporal derivative, right? So here again you have this response kernel measured in the group of Howard-Berg, right? So this positive lobe is basically it gives a positive weight to the very recent signal values in the past, and the negative is that it subtracts the signals further back into the past, right? And if you decrease the methylation time, basically this negative lobe moves towards the y-axis, and the system takes a more instantaneous derivative, a more recent derivative, right? And that's good, right? So if you take a more recent derivative, right, you take a derivative more recently, which has more predictive information. So that's good, but there's a trade-off, and that is that the gain goes down, right? Because what does this negative lobe does? It basically subtracts the signal, right? And so that reduces the gain. So if you instantly adapt, then your gain really goes to 0, right? And the only way to then beat that or circumvent that is by increasing the number of receptors and readouts, right? So increasing RT and XT, that increases the overall gain, but decreasing this methylation time reduces the gain, right? And so when the number of receptors and readout molecules is small, there's a lot of noise, right? So the way we would like to phrase it, this push-pull network downstream is a device that samples the state of the receptor. It really takes samples. And when you have a small number of them, you have a lot of sampling noise. And then the signal at the level of the receptor has to be relatively large, which requires a relatively large gain and a relatively larger methylation time. Whereas if you have more receptors, more readout molecules, this sampling noise is lower, and then you can afford to have a smaller gain, and then you can take, and that allows for a shorter methylation time, which then in turn allows for a more recent derivative, which is good for prediction, right? So, so being at the bound is very costly because the gain goes to zero, and the optimal system faces then a trade-off between a derivative that is informative, recent, versus one that is more reliable. Right? So a low tau m is a more recent derivative, which is more informative about the future, but it's less reliable because the gain is lower. This is the last slide, and then I think it's also time for lunch. It's a slide that I like very much. So what does it show now? So here the cost, RT and XT is fixed. So what we vary here is the steepness of the gradient, right? And then this red line is again, that corresponds to the optimal system that maximizes this predictive information under a resource constraint. So that's the system where we optimize over this ratio of receptors and readouts and this methylation time. The blue one corresponds to the E. coli system, where we fix the methylation time at what has been measured, and we also fix the number of receptors and readouts at what has been measured. Right? And what you can see is that when the gradient is relatively steep, right? So that corresponds to that dashed line at the top. Right? What you can see is that then the predictive information of the optimal system, marked by this red dot, is quite a bit higher than the predictive information of the E. coli system, this blue line, this blue dot over there, for G equals 0.4. And more interestingly, perhaps, that the past information is much higher in the E. coli system than the past information in the optimal system. So basically, E. coli uses a lot of, or has a lot of past information that it does not optimally use for prediction. And this is very different in this regime of shallow gradients. Now you see that these optima are very close together, right? So the predictive information of the E. coli system is very close to the predictive information of the optimal system. Moreover, now it uses this past information much more efficiently for prediction. Right? That's what this shows. Right? And so what we argue is that this chemotaxis, that this shows that this system is optimized to predict future derivatives in shallow gradients. Right? Because then it uses its past information most efficiently for prediction. Okay, it's time to wrap up, right? Right. So what we've shown is that these biochemical networks, they can, in principle, extract the most informative signal characteristics and reach the information bound. At least for these two classes of signals that we've looked at. Right? So these push-pull networks, they can copy the current input into the current output, which is optimal for these Markovian signals. And this chemotaxis system can take an instantaneous derivative, which is also optimal for predicting future derivatives. But at the same time, reaching this bound is very costly, right? So the chemical power diverges with a zero integration time. Your chemical power diverges, and in the chemotaxis example, we've seen that the gain then also goes to zero, right? And so in general, it's not optimal to be at the information bound, right? Cells can increase their predictive power per resource cost by moving away from it, right? So increasing this integration time reduces this chemical power and enables more measurements per resource cost. This is this idea of time integration. And increasing methylation time increases the gain. And then we've also seen that as this resource constraint is relaxed, right, and the system can have more receptors and readout molecules, that then the optimal system move towards the bound. And with that, I would like to thank you for your attention. Yeah.
So about the receptors, how much does the number of receptors you have, for instance, dictate like the maximum amount of information and the different paths that you can hold, and how do you think that would vary, like how much different environments this would, the number of receptors you have and the maximum path information you have?
Yeah. So right. So yeah. So basically, right. So if you look at this here, right. So here the constraint is this cos, which is the number of receptors plus the number of readout molecules, right? And as you relax this constraint, so if this sum is allowed to be bigger, then you also allow for more receptors, right? And so, yeah, and what this inset, what that then shows, that as you have more receptors and readout molecules, so you have a weaker resource constraint, that then this optimal methylation time becomes shorter. And then as such, you have a smaller amplification, but because you have more receptors and readout molecules, that compensates this effect, right? So the overall gain increases with how many receptors and readouts you have, and it also increases as you make the methylation time longer.
So in these kind of push-pull networks, if you have many readout molecules relative to the number of receptors, you enter this kind of nonlinear regime, right? In general, where the system can be oversensitive.
Yeah.
So I'm sort of two questions. One is, do I understand correctly that you focus on the linear regime, where effectively
Yes.
you're not there? And
yeah.
Then the question is, do you think it will change sort of qualitatively the results, or is it not relevant perhaps for coli, or I guess? Yeah,
so if the signal statistics is Gaussian, right, then the optimal mapping is a linear one. That's known mathematically. But of course, right, if they're very strong gradients, then of course the input can be distinctly non-Gaussian, and then nonlinearities may become important. But we've not looked at that. No.
So the impact also shows that when you increase the cost, the data is also increasing.
Well, yeah. So this theta is just a measure for this, how fast you or how far you are along this information bound. It's just sort of a metric that measures the distance along this line. So I think this theta as such does not have a deep physical meaning. It's just a way to parameterize the distance along this curve.
The ratio of x over r also changes the...
Yes, okay, so that I didn't show here. So the answer here is that it does not change so much. So for time integration, it's a very different story, right? So for time integration, if you make this integration time longer, right, I mean, basically explain this, then, right, so basically if you want to increase this ratio of the number of readouts over the number of receptors, if you want to maintain this regime that each receptor is sampled once every correlation time, right? That's because that's sort of the optimal thing to do, right? If you sample faster than once every correlation time, you take redundant samples. That's bad. That does not provide you any additional information. You're basically wasting readout molecules. But if you make it much longer, then your samples of the receptor are independent, but they correspond to all the samples. samples further back in the past, which are less informative about the future. So in the optimal system, you basically want to sample each receptor on a timescale set by this receptor ligand correlation time tau c. And then if you increase the number of readout molecules over the number of receptor molecules, if you would do nothing else, then each receptor will be sampled more frequently than this correlation time, which is bad. So to counteract that, you need to increase this integration time. And that you cannot do too much, because then you get these samples that are older, and this interplay gives rise to this optimal integration time. Yeah, but here we have this integration time fixed because we set this correlation time of receptor ligand binding to 0. And so here everything is set by methylation, and that then the optimal design is much less sensitive to the precise ratio, which is why I didn't show it. Yeah. Maybe first in the back? Yeah.
Can you clarify why there's no delta mu in this scenario?
Oh, that's because we wanted to focus on this, right? So we could have included that, right? But we know that basically that cost is of the same magnitude, and moreover, we've already demonstrated what happens then. So here we just really wanted to focus on this interplay between number of receptors and readout molecules and methylation time. Yeah.
This is a complicated question, so I'm not sure I can understand it myself. But there is an issue, right, when you do run and tumbles or any kind of change, that at the very beginning of a new behavior, the stored information is not particularly useful about prediction, right? If you just tumbled, I've got some information stored in PYP that was about my previous run, right? So as there's a kind of transition time.
Yeah.
Before that the information becomes more valuable. Okay, so that's the first part of the question yet. So the second part is, we just heard a talk where there was some delay where the motors don't switch at early times, right? I mean, you see some possible connection there that you don't, at least under some circumstances, it's not valuable to switch right after a previous switch because the information you've got isn't about your current trajectory.
So let me start with the second. Can I generalize this question?
Yeah.
I always wondered, I mean, the maybe simpler version is, why don't you reset the integrator when you tumble? Right? It would see, I mean, if you buy the general picture that, you know, you go straight and then you pick a completely random direction, anything you keep from before the tumble is totally useless.
Any feedback from the...
Well, no, I know, but you know, so...
Yeah, can you think of a system that does that? Yeah, but even more fundamentally, I don't know, right? So, so maybe one comment that maybe addresses this, and the first comment of Ned, right, which also connects to what Katja said, right, is that in many cases, right, this change in this angle direction is not so dramatic actually, right? So you keep going, you know, more or less the same direction. So I think that's one that's one important observation here. And the other question was about this delay, right? Whether that could be of any benefit, right? Or could there be a role, or what's the role of a delay more generally? That's a good question. So so there's two parts to this, right? So one is maybe something that Ben will touch upon this afternoon, I'm not sure, right? So, but basically in your paper with Machta, Henry, right? You basically derived the kernel that maximizes this drift speed. And this is in the absence of noise. And then you find that that's indeed an instantaneous kernel, right? So we basically find that the optimal kernel is a delta function. And that is indeed because in the absence of noise, this is indeed the optimal kernel, because the current derivative gives you the most information. So that then also maximizes this drift speed. Of course, what you did not look at was the effect of input noise, and that's basically what we have here, that if you have more noise, then it becomes beneficial to not have an instantaneous kernel, but one with a longer range, with a longer methylation time, because that basically makes it possible to lift the signal above this noise. You get a higher gain. Right, so here my answer would be that in the absence of any signaling noise, the best is indeed to take an instantaneous derivative. That's basically, I think, what you showed. But then what we show here is that then when there is noise, it's actually better to have a longer methylation time, because that allows you to lift the signal above the noise by raising the gain. Whether delay would play the same role, that I'm not sure. I mean, that you could think about, right? So maybe delay plays sort of a similar thing, like a sort of a integration time but no, I'm not so sure. I would have to think about that, yeah.
Maybe I missed this, but would you say that so the cost function has the sum of the receptors and the... Sorry? So the cost function has the sum
yeah.
plus xt.
Yeah.
Would you say there is an optimum ratio of these quantities as well, provided you fix the ligand binding time and so on?
Yeah, so right, so that's the first part of this talk, right? So with the push-pull network, right, there's an optimal ratio and an optimal integration time, which is this trade-off between maximizing sort of the number of bits of passed information, the number of XT molecules, versus how predictive these bits are about the future signal. And that's a trade-off, which gives rise to an optimal integration time and an optimal ratio.
It might be that interest for lunch is overcoming the discussion.
Yeah.
Despite the enthusiasm.
Yeah.
So let's thank Pieter Rein again.
Thanks.
So now with the correct title, among other things, I want to start just with some acknowledgments. So this work has really been led by Henry Mattingly, and it's been a really fantastic collaboration with Henry, Katja, and Thierry. Thierry is a common thread through all of this. And in the second part of the talk, or really the third part of the talk, I'm going to talk about a model that's really been developed by Derek and Isabella, and with a little bit of data that's starting to be taken for this project from Jeremy and Swayamshri. So to motivate this, we do know a lot about molecular details, biology's molecular details. So this is chemotaxis specific. And as we've heard before, the pathway for chemotaxis is really quite well worked out. There's some open questions which we'll talk about, but we really do have a lot of quantitative information about what the players are, how they interact with each other. And it's possible to take really high resolution measurements. So I'm going to be describing this in more detail, but what you're seeing here is data that Katja took looking at kinase activity. So the output of this, the output of Ka in individual cells, and you can see them fluctuating. These are in a constant background, but you can also measure response functions. And then on the other side, we also know a lot about high-level functions. So it's required to show this video or some version of it. And so I will do that. And they're doing run and tumble navigation, which we heard about earlier. And it's possible to write down quantitative models of this that are quite descriptive. And the goal, or my goal today, is to try and talk about an approach to trying to connect these two quantitatively. And it's sort of a reverse engineering approach. The goal is going to be to use information theory to connect the molecular details to the needs of a sensory, or to the needs of the behavior. So what we're going to need is a simple model and sort of a quantitative measurement of the behavior that these cells are carrying out. And we're going to combine that with some quantitative measures of the signaling fidelity. And the goal is really to understand sort of the design principles and what the constraints are that E. coli are up against. So to give a little bit of an overview, the first part of this, I'm going to be talking about connecting performance to signaling fidelity. And what I'm going to argue is that first, a fixed information rate places a theoretical bound on how well an individual bacterium can chemotax. So it places a bound on performance. Then I'm going to talk about measurements of this. So experimental measurements that really can measure how much information they get. And then we're going to compare their performance to this theoretical limit. In the second part of the talk, I'm going to compare the fidelity of the sensor itself to physical bounds. So there's been a lot of work to understand how the shot noise of individual single particle arrivals limits the ability of a sensor to measure concentrations. And we're going to extend this to understand how this limits the ability not to measure concentrations, but really to measure derivatives of concentrations. And we'll talk about how it seems like wild type E. coli, at least, are not terribly close to this bound, and we'll talk a little bit about why. And then in part 3, and hopefully we'll get there, we're going to talk about the mechanisms of signal amplification. And I'm going to argue that some recent experiments really strongly suggest some sort of critical lattice model. I'll go over what that evidence is. And also that recent work by people who are here really suggests that there's some sort of out of equilibrium mechanism involved in the coupling of receptors to kinases. And we're going to suggest a simple sort of percolation motivated model that we think connects these observations. Okay. So jumping into part 1. Okay, E. coli are not this interesting, or maybe that's, they are not this complicated, but our goal is to understand sort of the sensory information that's used in performing behaviors. And some of the challenges of this are more vivid here, that we're interested in understanding how much sensory information you need to control motor behavior. And information theory, in some sense, is measuring correlations. And there's also a lot of correlations that are driven by the feedback of motor behavior onto sensory inputs. So if the cheetah here is moving around, that's going to cause changes in its visual fields, and that's not sensory motor control. So we're going to have to distinguish that. And then what we're interested in asking is whether the information that their sensory system gets is needed and used efficiently in controlling motor behavior. Okay, so I want to give an analogy to image compression. So the question that we're going to be asking is really very similar to a rate distortion question. So you're most familiar from this, from images or maybe from film qualities. If I want to send you an image and I don't have a lot of information available to do that, I can send a low quality image. But if I want to send a better image, I have to use more information. And often this is written with the y-axis bad, so the y-axis being distortion. Then it's a rate distortion curve. But to make a better parallel with what I'm going to be talking about for chemotaxis, we're plotting image quality here. And notice there's a forbidden and allowed region. So you could be on this line, but you could also not be on this line. So in fact, I think the way that these images are saved, they're all the same amount of information, even though the one in the lower right is much higher quality. So what we want to ask, we're going to try and derive a curve like this and put E. coli on this. So we're going to ask how close they get to an analogous curve. I think I'm even embarrassed to play these movies given the earlier talks, but there's an animation, and here's the real thing. And to give an overview of this section of the talk, first I'm going to derive this bound. So this, on the left side, there's the drift velocity, so how rapidly they climb a gradient, divided by their run speed, v naught. And on the right side is a measure of the information rate in units of the rotational diffusion time. Then I'm going to talk about the information rate that real E. coli get. It's going to have this form. So the information rate, interesting. I don't know what that mouse is. Anyway, so, so the information rate is going to have this form. There's going to be a constant, and then the information rate is going to be proportional to the gradient squared. And we're going to derive that proportionality constant. And then we're going to compare real E. coli to this bound. Okay. So E. coli employ run and tumble navigation. Given the earlier talks, I can go through this very quickly. They have runs during which they move in a forward direction. And they intersperse this with tumbles where they randomly reorient. We heard a lot about the mechanics of this earlier. And they have some constraints. So they're 2 microns in length. The gradients that they want to climb are much larger than this, so 10 millimeters or so. And they also have rotational diffusion, so they can't go in a straight line for too long. And this sets sort of the maximum length that they could take runs for. So because they're too small to measure gradients across their body, they have to measure time derivatives, as we heard about earlier. And for us, the signal is going to be d log c dt. And they need to have sensory motor coupling. So if they don't have some way of sensing the ligand concentration, they can take a random walk. They can run and tumble randomly, but they can't move in a directed fashion up a gradient. Okay, so no sensory input. There's no gradient climbing. So to make the analogous plot, we're going to have drift speed now on the y-axis. So that's our performance, analogous to image quality. And now on the x-axis, there's going to be an information rate measured in bits per second. I'm going to need to tell you what this information rate is, but we want it to be a measure of how much the past of the signal influences the decision to make tumbles. Okay, so the first thing we thought about was the mutual information rate, and let me explain what this is. If I take 2 trajectories, so on the top, there's a trajectory of signal, and on the bottom, there's a trajectory of tumble decisions. And I could ask what the mutual information is between these two. And if I divide that by the length of the trajectory, I get a mutual information rate, something I learned about from a paper by Pieter Rein. And what I want to point out that you can sort of see in this particular simulation, there's actually no sensory motor coupling, so those tumbles are just random. But if you look at the signal plot, you can tell where the tumbles happened, right? Because that's where the signal abruptly changes. And in fact, in this case, that's because there's directional information going from motor behavior to the sensory inputs that is not what we're interested in. So what we want is instead the transfer entropy rate from the signal to the motor, and that's what we're going to call I dot from now on. So let me give a slightly more detailed model of chemotaxis here. So there's going to be behavioral parameters. This is a much simpler model. So these behavioral parameters are parameters that don't require information. You could put E. coli in no gradient, and you could measure these. And here they are. They're the rate at which you enter a run from a tumble. In our model that we've heard, this is not necessarily an accurate model. Signal does not influence that. So tumbles are exited randomly. The average run length or the average rate of exiting a run, lambda r. Alpha is a parameter that describes how fully reorienting you are. So in our model, that's also not something that they vary with signal, in contrast to what we heard this morning. And then they rotationally diffuse even when they're trying to go in a straight line, and they run with some speed v naught. Okay, and then there's also a functional of pass signal, which you require a gradient to sense. And so the idea is that the rate of exiting a run, lambda r, could be some arbitrary functional of the pass signal. And what we're going to try and do is choose a functional over the pass signal that minimizes the transfer entropy from signal to motor behavior while maximizing the up gradient velocity, if that makes sense. So, so just to specify this, if I give you this functional lambda r, you could tell me what the drift velocity is, and you could tell me what the information rate is, and together you could optimize this to generate the curve. Okay. So, okay, now I'm sweeping some things under the rug, but this is the form that this bound takes. So on the left-hand side, as promised, there's the drift velocity divided by the run velocity. Then there's some function of these behavioral parameters. That function is between 0 and 1. That's how the constants in the square root are decided. And then there's I dot over 12 dr. I dot is this information rate, and dr is the rotational diffusion constant. So this parameter is between 0 and 1. I guess the bottom of this is cut off, but it turns out measured from experiment. These are actually experiments that Henry did while visiting Katja. And this experiment gives an f of theta of 0.5. So they could do better by changing their behavioral parameters. And the main thing that they're not doing right is that they're tumbling too frequently. So this is something that Thierry has shown that if you just force them to tumble less, they'll climb gradients better. So this is not new to this, but in this framing, without getting more information, they could climb gradients faster simply by tumbling less. Okay, but so given those behavioral parameters, we want to know how close they can come to this bound. Okay, and before I get there, I just want to point out that the numbers on this are very small. So in fact, if you want to climb with say 5% of your drift of your run velocity, which is sort of typical in some experimental conditions at least, you only need something like 1/100 of a bit per second. So you don't need a lot of information to climb these gradients. And there's sort of a naive expectation that every tumble decision takes something like 1 bit. It turns out that their runs are about 1 second. And so to climb with a few % of their run speed, they really do need much less information than this. Okay, so far I've talked about how information puts a bound on performance.
Sorry, can you give us some intuition for, well, I mean, I guess constant out in front of the number, so that's hard to get. Why is it a square root?
Yeah, okay.
And is this some small trivial approximation?
Let's see. This is a small signal approximation. I don't know if this is quite going to answer your question, but one way to think about the square root is that the information is going to turn out to be proportional to the gradient squared. And so this bound has a linear approximation. So this does predict that the optimal strategy would have a constant proportionality between gradient steepness and climbing speed. This is making the small signal approximation. And I guess the other thing that I can tell you without really giving a calculation is that the optimal kernel takes the instantaneous signal, so it doesn't do any time averaging. And that's because the only way that in this model, the only way that pass signal influences or should influence the optimal strategy is if it has information about your current velocity in the gradient. Yeah, I mean, I guess dr is the only other timescale, if you will, in the problem. So I would say the I dot needs to be divided by a dr on those grounds. Yeah.
There's also a longer t, right, or a longer r.
Yeah, so those are in these theta. So that's in f of theta, and
How you partition the parameters between f and the
so the reason that I like putting it in terms of the dr is that's the thing that they really can't control, right? So they could change the tumble rate, they could change the run length, but rotational diffusion is presumably, yeah. And yeah, so theta is well, okay, dr is also in theta, but I don't know. Did that sort of answer your question? Yeah.
It's the thing that people like evolution can't really change much.
Yeah.
I was curious, is the assuming that we on some time scale where the reorientation is diffusive, could that also be a breakdown where it is not tumbling that much that essentially?
So this is the rotational diffusion constant while they're running. And you could also define a total rotational diffusion constant, and it turns out that's dominated by tumbles. And our optimum in this context would say that they should be equal contributions, actually, that the rotational diffusion and tumbling in the small noise approximation should equally contribute to rotational diffusion. But the measured parameters say that almost all of their reorientation is from tumbles. Yeah.
There's some intuition for why it needs so much less information to climb a gradient than you might naively expect.
Yeah, I would say when you're climbing a gradient at 5% of your run speed, you don't need to be biased very much in the up gradient direction, right? And so when you make a decision to tumble, you can be wrong almost half the time, and so the deviation of this transfer entropy is really quantifying how different the distribution of tumbles is if you know what the signal was versus if you didn't, and that doesn't need to be very much.
And I assume it's outside the regime of this approximation, but if you had a information rate of roughly 1 bit per tumble, or 1 bit per run, same, would the normalized drift velocity be order 1?
So this is outside of the regime of this approximation, but I do think so. I do think that I guess it's a little hard to say when it's order 1. It won't be 1, but my intuition would be it would be a half or so. Yeah, the question is so at 0.01 bits per run, they climb at 5 or so %. If you got up to 1 bit per run, how well could you do? Yeah, my intuition is also that the nonlinearities will make this lower, not higher, but.
Just to clarify that I really understand. If you say that the information rate is 0.01 bits per run, this is extracted from the tumbling behavior?
Yeah, so in this model, the only way that signal changes the behavior is by when runs are initiated. So,
so you're measuring the information rate that's encoded on the behavior, but that's not necessarily the same as the information rate that the bug is actually processing.
That's right. So this is if you could directly connect signal to the motor, and of course, there's many reasons that the real bugs might not get to this bound, but this is a bound for information that makes it to the motor. Yeah, and we haven't thought about what would happen if you could also alter, alpha, for example, with in the language of this model, I think you were saying that alpha is decreased by the good swimmers when they're going up gradient. We haven't thought about that. Okay, so now I want to tell you how we put real E. coli on this plot. And to do that, I'm just going to briefly go through this circuit that we've now seen a few times. So the receptors here, or the ligand rather, inhibits these receptors which talk to the kinases. So I'm going to talk more about how the kinases work in a bit, but for now, they phosphorylate Ky. Ky is really the only thing that talks to the motor, and it's dephosphorylated by Kz. And what's important, well, okay, so the things that really are crucial for understanding this, first, the system perfectly adapts. We've talked about this a few times today, so measuring the fraction of Ky, which is phosphorylated, and I'll explain how that works in a second. The plots that we've seen earlier are at the motor level, but if you look at kinase activity, what you see is that when attractant is added, kinase activity drops. So the bugs interpret this as they're swimming in the right direction. There's no need to tumble. But after some period of time, it returns to the same baseline. And now if you remove that attractant, you see a sudden burst of kinase activity, presumably because the bugs have interpreted this as now they're going in the wrong direction. Okay, as we've talked about before, Ki sort of represents a kernel derivative, right? So it's taking a derivative of concentration, so now rather than measuring the information rate to the motor, what we're going to do is measure the information rate to kinase activity. The question we're going to be asking is, if you took all of that information, so if all that information could get to the motor, how fast would they be able to climb, and how close are they coming to that bound? Okay, so in the low signal limit, the information rate can be written compactly in frequency space. If you could see here, there's a citation to Pieter Rein's paper where I learned about this, but it has this form. It's an integral over frequency space of a signal variance. So the amplitude of signal multiplied by some response kernel, and then divided by the size of noise fluctuations. And so now I'm going to tell you how we measure all of these. So K of t, this kernel, it's easier to talk about in the time domain rather. And this is the linear response kernel of kinase activity to a sudden change in concentration. So K of omega is just the Fourier transform of this. N of t is the autocorrelation of fluctuations in Ka activity in constant background. And then one of the things that's really nice about chemotaxis is that for many systems, we really can't measure like an ecologically reasonable signal statistics. But in a shallow gradient, at least, E. coli generate their own signal statistics, right? So they swim around, and in a gradient, they generate the signal that they see. So that's what we need to measure. And now I'm going to show you how we, and we is a combination of Katja and Henry, measured this. So we take advantage of a fret system. So this is how we measure the response. This is a really ridiculous microfluidic device. There's several channels there, which you can put different levels of ligand concentration in. Those are little cartoons of the bacteria, but they have this fret system in them. And by doing dose response measurements, Katja can measure in many individual cells the response to a sudden pulse of ligand. So here are many measurements here. You can average over individual applications of the dose response, and you can average over individual cells. And this is what those kernels look like. You can do a very similar experiment to measure the noise, now just not changing the ligand concentration. This is actually the plot that I showed earlier. There's a lot to take in these plots. They're interesting. There's a lot of cell-to-cell variability. But as far as we can tell, individual cells are pretty well fit by a single exponential. And the time constants vary cell to cell in an interesting way. And then to measure signal, we take advantage of this fact that it's really generated by the cell's own motion in a gradient. And so to measure this, you can track individual cells and measure their autocorrelation function in velocity. And in an exponential gradient of steepness g, the signal, as I've defined it, is just g times the upgradient velocity. And so these are individual tracks of individual cells. This is the upgradient velocity. And this, it's really striking that individual cells really have a lot of individuality here. So some cells take very long runs. Some take very short runs. And we take the average cell, or the average cell's parameters, rather than averaging these curves.
Sorry. If the autocorrelation function n of t is exponentially decaying, and the transfer function k of t has a roughly exponential behavior,
yep,
then computing this ratio actually becomes pretty delicate, right? I guess the question is, do you have a plot of n of omega in case for omega?
Let's see. In frequency space, I don't have this here. But yeah. Yeah, so you want k and n. This is n of t. Yeah. And this one here is k of t. So k of t is well fit by two exponentials, with one describing the rise. And actually that is probably faster than fret. So if I'm remembering the numbers right, if you change that from 100 milliseconds to instantaneous to a half second, which is the fastest fret timescale, there's a pretty small change in the information rate.
And the time constant of the decaying exponential is?
So that's one thing that's interesting. This seems to correlate very well with the time constant of the noise, and they're both, which makes sense. And they're both about 10 seconds. The methylation time, yeah. Well, yeah.
So that means that the ratio of response to the noise is almost frequency independent. If the response function is exponential and the autocorrelation of the noise is exponential and they have the same time constant, and that's equivalent to driving the input with a completely white noise.
That, yeah, I guess I do agree with this, yeah.
Multiply the response by the signal, right? And the signal has a different.
The signal has a different.
But if you think about, so you can think of the signal as being compared to an effective noise, which is the noise referred back to the response function. If those have the same time constants, then
yeah, as in, if I think I understand what you're saying, Pieter, Rein, the green and blue terms, those are the ones that cancel. The signal has a very different time constant, but yes, yeah, I think I agree. Yeah. Wait, sorry. So I do agree, but I haven't internalized the meaning of it. Okay.
If that's true, then the whole calculation is a little bit simpler. But it also is interesting because it suggests that, in some sense, your effective noise level is frequency independent. Right? Your noise level.
Yes, that's true. Yes.
Which means that over the full dynamic range you're talking about, the system actually isn't filtering anything because it's as good at measuring omega as 3 omega as 10 omega.
Yes. Yeah, I agree.
I guess intuitively that is because methylation is so much slower than the PYP, so the methylation noise is not filtered in any way.
Yeah, so
the YP, the phosphorylation dynamics, it integrates the receptor ligand binding noise. The methylation is much slower, also slower than phosphorylation, dephosphorylation. So that's basically not filtered.
Yeah. So yes.
I mean, I understand that you're going to end up in a different place, but if all the noise were the shot noise in the arrival of the molecules of the surface,
that would also be true. Yes. Yeah, that is true. That this is very consistent with that.
I think you're going to tell me you don't think that's what's going on, but
that's right. Yeah, not in these cells at least. But I agree. That is striking. Yes. Tentatively, yeah. Yes. Okay. So. Right, so these we're assuming are exponential. And out of this calculation is this rather simple beta times g squared. So this is the linear response. And just as a reminder, in this information rate, the signal is the autocorrelation of velocity. So that's measured in the absence of gradient, but the signal gets a factor of g squared. And so the information rate is beta times g squared, and the end result of this measurement is that they get something like 0.22 bits per second times a millimeter squared. Okay, and this is what that looks like. You can just plot this. It is quadratic in the gradient steepness. Okay, so our E. coli information limited. Now we can almost compare. The last thing to do is measure their climbing and gradients. And just to remind you what this measurement is about, it could be that this sensor is getting a lot of information, but it isn't being effectively used. And this could happen for a few reasons. Partly, it could be that the information that makes it to the motor is about past signal, but it's no longer relevant. And it could also be that the motor does not do a good job. So the motor could add additional noise And if that dominates, they would also be off this bound. And alternatively, it could be nearly saturated, and maybe it's easier to say, so this would imply energy is efficiently used if we define it this way. But what high efficiency means in this case is that all of the noise that gets to the motor is inserted by the kinases, if that makes sense. And the last thing we need to do is measure the drift speed in shallow gradients. Katja described the setup. I think Henry learned about it from her. But we measure, or Henry measures this gradient steepness. And the gradient steepness is measured at a bunch of different, the climbing speed is measured at a bunch of different gradient steepnesses, like so. And they have a linear response. And then from these two plots together, if you plot the y-axis versus the left, you can put bacteria on this plot. And they aren't quite at the bound, but they're pretty close. So about 2/3 of this. And as a conclusion of part one, I would say E. coli need very little information to chemotax to the ability that they do chemotax. But they do efficiently use the information that they do get. So they couldn't climb too much faster than this, given their behavioral strategy, without a better sensor. Okay, so I want to think of this as a slightly different information theory analysis, in that the goal is to understand, the goal is sort of to connect these molecular measurements to the performance of the behavior. Yeah.
I was going to ask you if it's possible to go to higher order gradients, but
yeah, yeah. So this does get sublinear eventually. So they certainly can't climb faster than their run speed, even in a steep gradient.
So would it be probably make sense to higher orders of gradients, and would that ever be relevant to study?
Yeah. So we have not tried to go beyond the linear case. It is interesting. I think Pieter Rein has some thoughts about this. Maybe I shouldn't. Yeah. Yeah. Right. So actually, I think maybe this was a question that Ned asked in a previous talk, that we ignore the fact that they could potentially know when their last run started. In the small gradient limit, I think we can ignore this sort of thing, because everything should be linear, or all of the signal should be linear. But in a steeper gradient, this wouldn't be possible. Okay. So I'll move on to part two if there aren't more questions. So far, I've argued that sensory fidelity places this bound on chemotactic performance of this type. And E. coli approach this bound. But what I want to talk about now is what limits the sensory fidelity. So what is it that sets the ability of the sensor to measure a gradient? And in particular, we're going to ask the question, how does stochastic particle arrival limit the possible information that the sensor could get? And just to show in this schematic, signal for us is deterministic. This is d log c dt, or a deterministic function of their position. But the cell only has access to the arrival of individual particles. And we want to understand how this sets a bound on the amount of information that they could get. And to talk about it in the context of this plot, we've been talking about the height of this and placing them on this plot, but now what we're going to do is understand what limits how far to the right they could be on this axis. Okay, and then we're going to ask whether E. coli approach this. Okay, so Berg and Purcell made this very nice argument about how the arrival of single particles, which have to arrive by shot noise, places a really fundamental limit on the ability of any sensor to measure a concentration in a finite amount of time. So each arrival is really stochastic. It's given by a Poisson distribution, so there's shot noise. And what they showed is that to estimate the concentration, if you have a time t, you cannot measure the concentration with a precision better than this. The fractional variance in concentration is given by 1 over 4 dA C naught t, or 1 over R naught t, where R naught is the rate of particle arrivals. And there's a lot of interesting follow-ups, I think many by people in this room, but I'm not going to discuss these. And instead, what we're going to ask is how does this limit the accuracy of measuring the signal, so the velocity. And actually, this is a very related question to what Pieter Rein was just talking about, just with a different noise source. Okay, so what can be learned about the signal from noisy particle arrivals? And just to make it clear what the question is, dc dt is given by c times what we've defined to be the signal. This is how the mean concentration changes with time. And the signal dt obeys these dynamics, so it decays exponentially with now the correlation time. So this isn't the rotational diffusion time, this is the total rotational autocorrelation time. And then there's noise, and the noise has this form, so the larger the gradient is, the larger this is squared, and so on and so forth, to produce the statistics that we measured previously. And now the assumption is that we're going to observe stochastic molecules at an arrival rate R of t, given by some R naught times the current concentration divided by C naught plus shot noise. And we're going to assume everything is Gaussian, so that the shot noise always takes the value that it has at C naught, if that makes sense. Okay, so as in part one, we're going to assume that the bacterium is just following the trajectory that it would follow in the absence of a gradient. And we're going to ask how well they could infer the current signal. And in particular, we're going to ask how much behaviorally relevant information they could get just by optimally counting particles.
Wait, sorry. So the statement that the bacterium follows the path that is the equation you wrote down, right?
Yes. Yeah, so given these statistics, how well, how much information could they get? Exactly. Okay, so I want to first lead you down a dead end that we started with, and that is to use the same formula for the mutual information rate as before, except now replacing this with the limits from shot noise. So the signal statistics would be just as before. The noise now, I think you anticipated this, Bill, is just going to be constant, so a constant in frequency, because the assumption is the noise is dominated by single particle arrivals, which are delta function correlated. And then the reason this is doomed is that the kernel now is taking an integral. So it's R naught divided by I omega, and the kernel really integrates the past signal. And so if you plug this in, what you see is that the mutual information rate actually diverges at low frequency. And this is interpretable. So the mutual information rate is really dominated by information about the position. This is about the past signal. So if you look at the whole signal trajectory, that's equivalent to knowing the position up to a boundary condition. But about the current signal, it's very different. Okay, so we need a measure that distinguishes past signal and present and future signal. So this is a path also very similar to what Pieter Rein was just showing us. So what we do is we construct an optimal causal filter to infer signal from particle counts. And a key parameter of this is what you could call a signal to noise ratio. So the signal to noise goes up when there's more particles, when the gradient is steeper, when you're traveling faster, and also with the cube of the autocorrelation time. And the optimal causal estimate of this takes the form of a filter, at least in the linear theory. So it's a kernel, and I'm just going to show you what the kernel looks like. You've probably seen this filter earlier. This kernel perfectly adapts, which means here that its integral is 0. It's always the sum of two exponentials. So a large and fast decaying exponential and a small negative but slowly decaying exponential. And as the signal to noise.
I guess I've lost the thread a little bit. The concentration as the time derivative of the concentration has exponential correlations because that
Yes.
the trajectory.
Yes.
And your job is to observe a Poisson process whose rate is the concentration.
Yes.
And your job is to estimate the
current velocity.
Current derivative from the current time derivative of the concentration.
That's right.
From, okay.
Yes. So the shape of this filter depends on the signal statistics, which we're again assuming are exponential. If that's okay. So, okay, hopefully this will help. The predictive information here is the mutual information between your estimate of the signal, which means your convolution of this, or sorry, multiplication of this kernel with the past history of single particle counts. It's the mutual information between that and the actual signal, so whether you're actually moving up or down. That is what you could call the predictive information. This is how much information the past history of particle counts gives you about the current signal. And it turns out that in the small gradient limit, it has this form, so 1/12 R naught. So it increases as you increase the rate of single particle counts, and the gradient squared, much like that. And this isn't quite what we need here. What we need exactly is what I want to call the predictive information rate. It's how much less you know about a little time step dt in the future versus how much you know now divided by dt. So it's sort of how quickly does your information about the future decay. And this is the form it takes in this problem. So, okay, so this was an elaborate calculation that I skipped a lot of intermediate steps on, but the end result is that this is how much behaviorally relevant information you could extract in bits per second if you could count individual particles as they arrive at the sensor. Okay, maybe I should give some intuition for this. So this increases with R naught, and also with how how large your signal statistics are. So that's the gradient steepness times v squared. Okay, so that is the theoretical bound. We can also measure how much information they get. I almost showed you the experiments needed for this, but I will just explain what's going to be different about this. So again, the things that we need to measure the same information rate is again the kernel and the noise, but now at much lower concentrations. So the measurements in the earlier part were deep in the log sensing regime, so at very high concentrations, and cells are most likely to be single particle counting limited when concentrations are very low. And I'll explain in a little bit why these particular concentrations make sense, but these measurements are repeated at 0.1 and 1 micromolar. And a first hint that they might not be shot noise limited is that the noise does not depend strongly on the background concentration. So here is a plot of the kinase activity of 2 cells at 0.1 micromolar. And if you look at their correlation in the time domain, they look pretty similar. There isn't much of a trend between these. If you look at 1 micromolar, these plots look similar, and they look similar to the ones taken at much higher concentrations. Okay, so to compare to the bound, I need to give some numbers. So R naught, the parameter in the theory, is 4dAc naught, and it's some constant times the concentration, which we'll call Kd. And for a sensor, we're going to assume the sensor is 60 nanometers. This is the diffusion constant of aspartate, and this gives a Kd of something like 10 to the 5 per micromolar per second. We have the same measured parameters as before, and this gives this theoretical curve, which always has slope 1 on this. So this is plotting the background concentration on the x-axis, and on the y-axis, there's the information rate per gradient squared. So this is the thing that depends on the rate of single particle arrivals. Okay, so that is the black curve. The blue points here are measured from experiment at 3 concentrations. The curve is an interpolation between them using the MWC model and assuming this constant noise that's been observed. And let me give a little bit of intuition for this. So in the MWC model, there's a log sensing regime in which the response is to a change in log concentration. And in that regime, the information rate is constant. And that's because the signal is constant and the noise is constant as well. Okay, and it also has a regime in which the response is proportional to the absolute change in concentration. So at very low concentrations, they cease to be log sensitive and respond to absolute changes. And in this regime, the response to a change in log, which is what we're plotting here, is always proportional to the concentration. And so in the MWC model with this noise, in this regime, the information rate goes as the square of the concentration. So a different slope. And plotting these a different way, if you look at the ratio of the information rate to the actual kinases to the bound, this has a peak somewhere near the bottom of the log sensing regime. And if you look at the y-axis of this, it's about 1%. So at least these cells do not appear to be particularly close to this fundamental limit. Sorry? There's 3 points, so we would like to take more. Yes, that's easy for me to say. But it is true. This does appear to be a wild extrapolation from these 3 points.
There's a far away from this again because it's dominated by methylation.
Okay, so I have some thoughts about this. So I have a few possibilities. The first is that these are wild type E. coli, and they have to sense multiple ligands. And here they're only seeing aspartate, and all the other receptors are sort of dead pixels, right? In this paper from 2003 that you can't quite see the citation of, from Sorgic and Berg, they showed that if you instead make mutants that only have TAR receptors, they have a several times higher gain and increased sensitivity. So they respond to lower concentrations. They also have higher noise, but it's possible that that mutant would get much closer to the bound, probably not 100-fold higher, but the gain is squared in these. So this seems plausible. The other possibility that I think you're hinting at is that maybe there's just other constraints. In particular, in the log sensing regime, if you assume that they are counting signal molecules, they're counting about 10,000 per second. And maybe this is pretty impressive, and maybe we shouldn't be worried about why they aren't counting 1 million particles per second. We should instead be thinking, how is it that they can really count something like 10,000 molecules every second? Two more specific things within this, you could imagine that the amplifier, so the signal needs to be amplified, and you could imagine that the needs of the amplifier just introduce a lot of noise. And you could also imagine, as you were saying, that the adaptation mechanism introduces noise. And then the third possibility is that there's other behavioral demands. So it's possible that the two bounds that I've just presented can't be saturated at the same time. So the first bound could be saturated, but it's possible that if you have a signal that saturates this bound, that's not behaviorally useful information necessarily. For example, you can imagine that in transforming from this continuous signal that you'd get from the optimal kernel into discrete tumble decisions, you'd lose enough efficiency that it doesn't pay to increase the information rate at the sensor.
Sorry. Isn't the formula that you start with, where you write an information rate as an integral of the signal-to-noise ratio, only valid if the signal-to-noise ratio is small?
Yes.
But although it's small for the kinase, given the numbers that you're plugging in, it is not small for the receptor shock.
Yes, that's true.
Which means that there's a big difference between 10,000 and the log of 10,000. Right? So the general formula has something like log of 1 plus the signal-to-noise ratio.
Yeah.
So you might be wildly overestimated by using the low signal-to-noise ratio formula and applying it to the shot noise. You might be wildly overestimating the information that's available from the initial measurement.
Yes, I do think that. Let me say that in the integral formula, that diverges, and if you replace the linear approximation with a log, that would absolutely not diverge, but you wouldn't get a term proportional to g squared. So you wouldn't, the linear response is not well defined, or the low signal. But I do think that the predictive information rate that we've calculated is not sensitive to these in the same way. It's in the time domain. Yeah. Yeah. I guess my main evidence for that is that it doesn't diverge in the small gradient limit. But okay, so just to recap this, I've argued that, or shown you how the shot noise of single particle places some bound on the accuracy of gradient sensing, as opposed to, or gradient sensing through time derivatives, as opposed to of measuring absolute concentrations, and argued that measuring velocity is very different from measuring position in terms of information rates. The bound very heavily depends on the behavioral strategy, and at least in this experiment, with a lot of caveats, wild-type E. coli don't seem to come very close to it.
Can I ask a question? I lost where this is, but I'm wondering about this business that this run and tumble strategy is in some ways special, because you don't really care about the diffusive part of that. Like, you do runs and tumbles, and if I just added, let's imagine that I had a tiny extra distance when I ran up the gradient and down the gradient every time. That's great. But I could also just randomly add additional distance up and down. But if on average, every time I moved up, I moved a little farther, I would still have a good drift velocity, right? I would.
Yeah.
So there's, in some sense, I could add a lot of noise to the system, but as long as I'm preserving the signal and it's unbiased between, and it keeps the bias between up gradient and down gradient, I still perform. Yeah.
So I have a precise answer to that, which is that would have a lot of information, right? Because you know if you take a 1.2 second run that you're going in the right direction, whereas if you take a 0.8 second run, you're going in the wrong direction. So the optimal by this very specific criterion of maximizing the drift velocity while minimizing the information rate does say that you should have in the small gradient regime, says that you should have a Poisson rate that's biased by what direction you're heading in, as opposed to this other strategy of going a little longer if you're going uphill.
In some sense, the actual variation doesn't matter. I mean, the overall noise in run distance doesn't matter so much, just the difference between up and down. Like, I could have these two distributions,
yeah,
and I could have them be very, very broad and huge and large runs, but as long as the difference was there, I would still manage to move up.
I don't have a great answer to this, and it's a little surprising that in converting from this continuous signal to a discrete one, there isn't more information lost in the sense of looking at the key Y signal and performance. Maybe an answer is that because it's so noisy, the amplitude is really saying how confident you are in whether you're going up or down. And so I don't know if that quite satisfies you, yeah. But I would say that they're this close to the bound, I think really does imply that there's very little noise left at the motor. So if you know what the key Y concentration is, that tells the motor what to do, and the noise is upstream of that. Okay, so what I want to spend the rest of my time talking about is a model for the signal amplification. And this is work that Derek has been leading with Isabella. And so I want to just talk about the coarse functional features of the bacterial chemosensory array that need to be explained. So the first is that this array has very high gain, very high effective cooperativity. And what you're seeing here, I believe this is data Swayamshri took. This is in a similar microfluidic device. On the top here, you're seeing a protocol of concentration pulses. And And in these panels, you're seeing individual cells' responses, and you get very clean data. And from this, you can pull out a distribution of cooperativities among cells. And in this experiment, you get a range of 5 to 20 or so. So this is very high cooperativity. The other thing that we've talked about is perfect adaptation. So I won't dwell on this more, except that if you give, at least in a log sensing regime, if you change the background concentration, you get a transient, and then you return to normal. And then the other thing that's very striking is if you look at an individual cell that's not seeing any signals, you see very large fluctuations. So this axis goes from 0, which is if you fully inhibit the kinases, what the signal is, to 1. And the point that I'd like you to take from this is that the fluctuations are order 1. So even though there's a few thousand receptors and kinases, they seem to all be fluctuating together. Okay, the structural features of the bacterial chemosensing array are also very striking. So what you're seeing here is a side view. This is the membrane here. The receptors go through the membrane. There's 6 receptor dimers, there's 2 trimers of dimers that form a core complex with 2 kinases. The kinases are key A, and 2 structural proteins, or maybe proteins of unknown function that form this core unit. And this is what it looks like from the top down. But maybe important to note that these are quite spatially distinct, where the business happens for the receptors and for the kinases. That's one core unit, but the core units are organized into a superstructure, which looks something like this. So now the orange here are these kinases key A. The light blue is the structural protein key W. And they form actually a Kagame lattice. A Kagame lattice with a very large unit cell. So very complicated lattice structure. And this is a cartoon, but this is really cryo-EM data. So this really is a lattice in cells. It reorganizes. There's a lot of interesting complications, but they're organized into a lattice. And if we zoom in on an individual receptor, most people think of these receptors as being equilibrium sensors, where the probability of being on, so often referred to as active, is given by 1 over 1 plus e to some change in free energy, where this is the free energy between the on and off states. And typically, this is written as some constant plus a constant times the number of methylation marks, and I'll explain what those are in a moment, plus a term that depends on ligand concentration. So this receptor works because the 2 states, on and off, have different affinity for ligand. That's what this term here quantifies, and the log sensing regime, with a bunch of caveats, roughly goes when the concentration is between the affinities of these 2 states. And the methylation level reweights the relative states to keep the receptor in the range where it's roughly half bound by ligand. So I put an asterisk here because this may not be entirely true. But to give the usual picture of perfect adaptation, these enzymes that add and remove methyl marks can have very simple dynamics where when a receptor is off, key R can add a methyl mark, which increases the probability of being on. And when it's on, key V removes a methyl mark. Okay, so this is a single receptor, but getting this large gain or this large cooperativity requires some coupling between them. And I just want to give, a little bit of an overview of the 2 classes of models. so one type of model or class of model is an MWC type model. And in the MWC type model, there are many receptors in a tight cluster, a signaling team perhaps, that all need to transition together. If they all transition together, then the probability of finding them in the on state just gets this factor of n, where n is going to be the cooperativity that you'd observe in an experiment. And MWC models are excellent phenomenological models. They essentially fit all of the available data, with one potential caveat that they don't really naturally fit this noise. So these clusters do increase the noise, but there's still a lot of clusters. So to get a cooperativity of 20, you'd still need many, many receptor clusters, and presumably they would be independent. Okay, so the other class of models are Ising coupled models. And here, receptors are just coupled to their nearest neighbors. And there's some energetic cost to having different states. And here you have a large effective cooperativity as long as this Ising lattice is held very close to its critical point. So Ising models very naturally explain sort of the structure, the superstructure of these in a lattice. And one thing that I find particularly appealing is that they can explain the noise as arising from proximity to this critical point. So in this picture, all of these receptors are coupled together into one big lattice. And you could imagine a fluctuation would push many of them together. Okay, so let's see. So that was the state of the field, I think literally until 2 months ago, the published field. And there are two very exciting recent developments. The first is this paper, Kijkstra et al. From Tom Shimizu's lab. And what they showed is that if you take mutants that have the adaptation machinery knocked out, and I believe also have just a single receptor type, that most of them will be either all on or all off. So their activity doesn't change much in the experiment, but a subset of them will transition rapidly, roughly between being all on and all off. And they show this for a few different receptor types. And you can see some cells that are mostly on but occasionally switch off, some that are mostly off but occasionally switch on. And they argued that this really strongly suggests some sort of coupled lattice model. And they interpret this as being consistent with an Ising model that's very close to its critical point. And I would say this really strongly suggests that somehow there's coupling through the lattice and that that's how the amplification happens. But there's other recent evidence from David and Chiwei, who are here, and Yuhai too. And this is actually from an older experiment, I think from about 10 years ago, but a new interpretation of it. So what you're seeing here is kinase activity, a dose response curve of kinase activity in two different methylation conditions. So these, there's no adaptation machinery. And so you can measure a dose response curve, and as expected, methylation shifts the dose response curve as we all know it should. But if you look at ligand binding, which is much harder to do, I actually don't know how this works exactly, you see that the ligand binding hardly shifts with methylation. And this is very striking. What you're seeing here are the data, and these curves are the prediction of an MWC model that's fit to the kinase activity, if that makes sense. And what I think you should note, the receptor binding is a little bit less cooperative, but it really doesn't care about the methylation nearly as much as the kinase activity does. And this really suggests that the kinase activity is not simply an equilibrium with the state of the receptors. And they interpret this in a model where methylation alters some sort of kinetic rate that's a barrier to kinase activity. Fair summary. Okay, so these are the key phenomenology to explain. There's very large gain, a lot of cell-to-cell variability in that. There's perfect adaptation. There's large fluctuations, and in certain conditions, two-state switching, even without the adaptation machinery. There's decoupling of receptor binding and kinase activity. And then one thing that I find very appealing, but we could debate whether this is a key component, somehow this shouldn't have a lot of fine-tuning. Maybe. I think opinions vary on this. You could imagine that this is one number, and cells could tune something like an Ising coupling. But my personal tastes are to look for a model that doesn't have fine-tuning. And that's actually, we weren't aware of a lot of this data when we started developing this, but we do think the model that I'm going to present to you does a pretty decent job of capturing this phenomenology. Okay, so a very broad motivation for this is to think about bond percolation. So in bond percolation, there's a control parameter, which you can think of as the fraction of bonds on a lattice that are occupied, and an order parameter, which you can think of as the fraction of sites that are in the largest cluster. And as you go through a percolation threshold, as I change this control parameter, it goes from 0 to non-zero very steeply. So very close to this, there's a diverging susceptibility of the order parameter to small changes in the control parameter. What we're going to introduce here is a model where the receptors are something like the bonds and the kinase activity is something like the order parameter. Okay, so that's the motivation, but here's a specific model. So importantly, receptor state is different from kinase state here. The kinases live on the vertices. They can be active or inactive, and the receptors live on the bonds. This is a square lattice for simplicity, and they can be off or on. And the dynamics are as follows. Activity and inactivity are going to spread through receptors, but they're only going to spread, activity is only going to spread through on receptors, and inactivity is only going to spread through off receptors. So if we zoom in on two sites here that are connected by an active bond, then there's some rate that the activity will spread from this site to that one. And alternatively, if this bond is inactive, then there's some probability that inactivity will spread. Okay, in addition to those dynamics, we also need that key A will occasionally spontaneously activate or deactivate. And this is so that the system doesn't get stuck in a trap and to make the dynamics faster, as we'll see. So for some intuition for this, imagine that this spontaneously activates. It can spread to all of its neighbors that have active bonds or on bonds, but similarly, inactivity can spread like this. And as you can maybe see from this, the fraction of kinases which will be active is very sensitive to the fraction of receptors which are on. Okay, so I'm going to show you a mean field solution to this. So A is going to be the mean kinase activity. P is the fraction of on receptors. Z is just the lattice coordination number, so 4 for a square lattice. And K is this rate of spontaneous transitions. So in this, we have dynamics like this. Inactive receptors can become activated at a rate that's proportional to the number of active neighbors they have, times this factor arising from methylation, but they can also spontaneously activate. And there's a similar dynamics for inactivation. And if K over P is small, there's very high effective cooperativity. So this is plotting the steady state kinase activity, where on the x-axis now there's p e to the alpha delta m, and on the y-axis there's the mean kinase activity. And what you can see is that when k is not so small, so when there are many spontaneous flips, this is somewhat steep. But as k gets very small, this becomes a very steep function of the fraction of receptors which are on. So this gets its cooperativity really by being very close to this percolation transition, which happens at p of a half in percolation, but when this thing is a half in this model. Okay, so I haven't told you about adaptation yet, but here Kr is going to be the rate of methylation by Kr. Kb is going to be the rate of demethylation. And in mean field, we get equations of motion like this. So methylation dynamics have two important parts. So firstly, only receptors which are off can be demethylated, and which are on can be methylated. But now importantly for this model, and actually less importantly for other models of adaptation, it's very important that Kb gets feedback from the kinase, and that's to make sure that this tunes to the critical point. So let me explain this. Well, anyway, this is the mean activity, and the mean activity now depends. This is the DMDT nullcline. So this is the nullcline where there's no net change in methylation. And now this is a function both of the probability of an on receptor and the probability of an active kinase. And putting these two together, what you can see is that the fixed point is given when both the activity dynamics are dAdt equals 0, or the crossing of these two nullclines. What I want you to note is as this orange curve gets very steep, that's what happens when the model without adaptation is very close to this percolation transition, then whatever the dp/dt nullcline is, it'll almost certainly hit it very close to its critical point. Yeah, the argument is this orange curve here is dAdt equals 0. And when k gets very small, which I would argue doesn't require fine tuning, right? That's just saying there's a separation of scales between these two processes. As that gets small, this gets very steep because it has a percolation transition and basically goes from all off to all on. And as long as this other curve depends on kinase activity, it will cross somewhere at that transition. That's the idea. Okay, so this model perfectly adapts, but it doesn't just perfectly adapt, it perfectly adapts to this critical point. Maybe you would want to call this self-organized criticality. And now the system does respond with a very strongly amplified transient, because if I add some ligand, that shifts me over this way in P, and because there's this very strong amplification, that leads to a large change in activity that then gets reset back to the same fixed point. Okay, so without going into a lot of detail about this, simulations on lattices have large fluctuations when k is small. These are really critical fluctuations. They're there if you tune the system to the critical point even without the adaptation machinery, and they persist with a different form when there is adaptation machinery. And when k is very small and without adaptation, we have two-state switching, but maybe let's not dwell on this. We haven't really thought about the parameter regime. but it is a very reasonable prediction of a model of this type, where all of the sites are strongly coupled. So just to recap this final part, a percolation model really requires, well, it gets its high gain very differently from cooperativity by being very close to this percolation critical point. We recapitulate these large fluctuations and the two-state switchings, and here they're manifestations of critical phenomena rather than driven by the adaptation machinery, although they could be both. This somewhat naturally explains the separation of receptor binding and kinase activity. And it doesn't really require fine-tuning. It does require that this ratio is small, and that is sufficient. And I would say this is different from the Ising model in this way. So these other things do mostly apply to the Ising model, but the Ising model does require fine-tuning of J. So the adaptation machinery can tune the effective field at the receptor level, but not the coupling. Okay, so just, I guess, I have passed my time. So why don't I thank my group and collaborators and funding and take questions. Ned?
Yeah. So one thing that I'm wondering is, can you explain the very large range over which, say, cells respond to a aspartate or methyl aspartate? Because that, I always understood it as very different values of the binding affinity, and then that being coupled to changes in the on and off states.
Yes.
In an equilibrium sense.
So if I can reframe your complaint, like this plot seems very mysterious in that they really can respond to aspartate, you know, over a good chunk of this range.
5 orders of magnitude.
Yeah, and in the MWC model with methylation changing receptor affinity, this all makes sense, right? You have, you know, the upper range is set by the affinity of the high affinity state. The lower range is set by the affinity of the low affinity state. Because methylation doesn't shift this very much, I think that really strongly suggests, firstly, that the cooperativity can't come at the receptor level because, you know, how could you be cooperative when when an individual receptor is bound 10 to the minus 2 of the time? But it also suggests that you need to somehow be able to sense both when 1% are unbound and when 1% are bound. So it wouldn't be sufficient to just have a response when a receptor is bound or just when it's not bound, right? There needs to be something that happens that's detectable. So 99% needs to be different from 98%, but also 1% needs to be different from 2%. We sort of put this in by hand here in that the methylation dynamics change both the spreading rate and the spreading of activity and the spreading of inactivity. But I agree, it's a mystery, and that part does feel a little unnatural.
Yeah, can you repeat how the methylation, right? So you have this equation for methylation, it depends on the active and the inactive state, right? So, and then also on the activity of the kinase, right? And that's important.
Yeah. Okay, so Kb gets phosphorylated, and this increases its activity quite a bit. If this constant Kb didn't depend on the activity, then this would be the picture where perfect adaptation just happens by only methylating off receptors and demethylating on receptors. And for an ising-coupled model and an MWC type model, this is enough. But it isn't for us because if there's no dependence on kinase activity, this orange line here would be vertical. And so it actually wouldn't be natural for it to go through the critical point. And so it doesn't need to have much dependence on kinase activity, but it can't go exactly vertical, if that makes sense. So
probability for bond, and that the receptor acts as a bond is P, which is in that equation, right? So that depends on the ligand concentration, right, really? But does it also still depend on the methylation level?
This is an excellent question. So we only recently became aware of this these recent results about how methylation doesn't change receptor binding. And the way we've put this in, which I think you could complain about, is that it has the same factor of e to the alpha m. It is a problem for this model if it only cares about whether the receptor is bound or not.
Yeah, so I know that in model of UI also, right, that the methylation no longer changes the relative stability of these states. But that by itself is also somewhat of a mystery, right? If you add chemical group to the protein, and that changes its free energy in general. And now you add this group, and that somehow magically does not change the relative free energy difference between these two states.
So
it can happen, you cannot rule it out, but it's also a bit mysterious. No.
So I think of it, in this model, I think of it as changing the rate at which activity attempts to spread to a neighbor and the rate at which inactivity attempts to spread to a neighbor, if that makes sense. And in my understanding, it's very similar in their model, except their model is just for a single receptor, in that it's some intermediate state that they think of methylation as destabilizing or stabilizing, and thereby changing the rate at which you hop over some barrier. Is that nodding is a little less vigorous? Yeah. Yeah. Yeah, I mean, I think I agree that data is very hard to explain without something like this. Yeah.
I'm wondering how general the idea that we have is here. So if I think about, I mean, what you're saying is that if I need the intersection of two null lines and one of them is a step function,
yes,
then I'm going to hit the middle of it.
Yes.
Actually, it's generic that I hit the middle. It would be very rare for it to be non-generic to hit the edges.
Mm-hmm.
So can I think about some much more abstract thing where I have, you have some particular mechanism that generates these things, but is it more general than that? That if somewhere in parameter space there's a place where you're almost step-like, then if you have some adaptation scheme, then generically you're going to go there because.
Yeah, I think this is, so we're exploring a few other examples of this actually. I do think this is a good way in general. So really, Isabella's working on a few things where this is the common thread that if you have, there are subtleties. So if it's a bifurcation, then maybe this doesn't work. So it needs to be a step function without hysteresis, which maybe is a little unnatural.
I guess the other question is this something, you've talked about this in a mean field way. So you're sort of mixing the sort of critical phenomena, which have some ends going to infinity.
Yes.
And sort of, something happens and you end up with a sort of dynamical system with two null points. So should I mean, is there some question that's analogous to whether, disorder is relevant at the critical point? Since you can't, or would you argue that, in fact, the coupling which adjusts things itself has a kind of neat field?
So, okay, I don't know if this is quite your question, but I do think that it's important that the adaptation machinery not only gets feedback from the kinase, but also is specific to methylating off receptors and demethylating on receptors, so that you don't get dead pixels, if you will, right? That
right. So I can imagine an adaptation mechanism that's sensitive to the integrated activity of the entire. I don't know whether this is actually how it works here, but
yeah.
if I think about the more general case, it's not so hard to imagine that there's an adaptation machinery that knows, the total magnetization of the lattice, right?
Right.
But what it does is it goes in and adjusts the local fields at every site.
Right.
And so you might worry that disorder in those local fields disrupts the critical behavior.
Maybe Derek actually knows more of the latest than.
There have definitely been having sort of a bunch of variability in the local field definitely does lead to some interesting messiness in the critical behavior that I haven't fully understood yet. But there is definitely some messiness there where when you have this disorder in the local field, the critical behavior is not quite as sharp. I think it's fair to say.
Okay, let's thank Ben.
Thank you.